Thank you very much for inviting me here today. And it is my pleasure to present our research on the analysis on fungal microbiomes associated with the soil, root, and nodules of the food quality winter pea in the Pacific Northwest, USA. Why we are focusing on dry peas? Dry peas is a very important crop in Washington agriculture, with a production value close to 15 million US dollars. This crop occupied at least 27,000 hectares and grown in rotation with the small cereals. There are two big groups of dry peas, spring peas and winter peas. Spring peas mostly used for the human food and are planted in early spring with the crop production in middle of the summer. The winter pea are cold tolerant. Because of their cold tolerance, they can be planted in the fall, September, October, and they can survive of the winter, as a result producing crop in early summer. Until 2009, the primary use of winter pea was cover crop or animal feed purposes. However, in 2009, there was a changes in the regulation allowing some cultivars to be used as a food-grade winter pea suitable for human consumption. And after this, these new cultivars become very popular with growers and attract a lot of interest in the research. But before I start to talk about fungal community associated with winter pea roots, I would like to talk about more general feature of winter pea as well as other legumes, abilities to establish symbiosis with soil bacteria rhizobia. On this figure, you can see a section of the nodule formed on winter pea by rhizobia, which we label with the protein which is expressed in symbiosis, which allow to visualize nitrogen-fixing cells inside the plant tissue with the green color. And this is a second picture is just nodules showing on the plant roots. So this ability of legumes to fix symbiotic nitrogen and symbiosis is very important in sustainable agriculture. It's helped to protect environment from pollutions and nitrogen contamination. It's also provide an alternative way to serve energy and as well conserve ecosystems. In addition to general or direct effect of fixed nitrogen, rhizobial symbiosis also can promote plant growth and stress resistance. As a result, this symbiosis can improve plant resistance to biotic stresses, which become more and more important in climate changing environment. As well as the symbiosis can improve crop nutritional quality. Over a long history of winter pea used by human, legume domestication result in the significant changes, especially shown in newest modern cultivars. For example, the new cultivars become more dependent on soil nitrogen than to symbiotic nitrogen fixation. This selection also alter the ability of plant to interact with bacteria and fungi, and also affect the ability of the plant to select of highly efficient rhizobial partners as well as to resist diseases. Additionally, we should remember that rhizobia are not the only habitats in the legume nodules. There are a number of other microbes, bacteria, and fungi which occupied internal part and external part of the nodules. These microorganisms can play plant growth promotion function, promote or prevent establishment of effective symbioses, improve or decrease nodulation, and could result in the development of nodule disease, which also can affect symbiotic nitrogen fixation. Additionally, because these winter pea cultivars, food-grade cultivars, are very new, even less is known about the microbiomes associated with these plants. So the goal of our research was to assess the winter pea root-associated pathogens, to understand the diseases, to be able to protect the plants against these pathogens. We also want to understand fungal community in winter pea nodules, more specifically to assess potential fungal pathogens enriched in the winter pea nodules. Again, to see if we could target these pathogens to improve symbiotic nitrogen fixation, and also evaluate a positive effect of the fungi on the nitrogen fixation. And most of this research is very new and very little known about these questions in any legumes. In our research, we evaluated the plants grown in the different locations across Pacific Northwest and analyzed microbiomes across different tissues such as nodules, roots, as well as soils. And also incorporated in this research number of plant genotypes, which allow us to look at the interaction between plant genotype and environment. We also look for interaction between plant genotype and the effect of environment on the plant performance, looking for growth parameters and symbiotic performance. Most of the research which I'm going to talk about today is focused on the metagenome sequencing, which incorporate long reads and short read amplicon sequencing. But in the future, we are planning to incorporate much more comprehensive analysis, including metagenome and some custom amplicons. So far, our study incorporated numerous locations across Idaho, Washington, and Oregon, and at least 10 different winter pea cultivars. However, for this research which I'm presenting here, I will be focusing only on the analysis of the 3 winter pea cultivars grown in 2 locations, one in Idaho and one in Washington. In the study, we evaluate the nodulation and disease development of the plants and assess fungal microbiomes using short-read ITS amplicon sequencing, as well as PacBio ITS amplicon sequencing. The PacBio sequencing was used mostly to narrow down to specific taxonomic taxa of interest. As a result, we identified close to 100 different ASVs in this microbiome. We assessed alpha diversity and beta diversity of this community, evaluate community composition, and look at the co-occurrence of the different fungi to see, to find some specific complexes which can be used in the future to assess pathogen co-occurrence. In the study, we identified close to 1,000 ASVs in combined root, soil, and nodule microbiome. We assessed fungal alpha and beta diversity and also community composition, comparing relative abundances of fungi between soil, root, and nodule environment. We also analyze co-occurrence network to identify specific fungal complexes which coexist together in winter pea roots. So here are some figures showing some of this analysis, but because of the time limitation, I'm not going to focus on the general description of the microbiomes, and I will go directly to discuss the specifics in the differences in the community profiles. First of all, let's start with the basics, the foundation of plant-associated microbiome, soil microbial communities. In both locations, we identify a number of putative plant pathogenic taxa which were present in high abundances. We also found a significant variation in relative abundances of these pathogenic taxa between locations. For example, Didymella was highly abundant in one location on your right in Garfield and was represented by 16% of total ITS reads in this location compared to 2% in the second location. Alternaria, on the other hand, was highly abundant in Pullman location, but much less represented in Garfield location. Fusarium also had significant difference. This is all three known pathogenic genera containing a number of P pathogens. Next question we ask, what are the fungal genera which were significantly enriched in the winter pea roots compared to the soils? So in general, the most abundant and important taxa which were overrepresented in winter pea roots across both locations were Rhizoctonia solani, Didymella, and a number of arbuscular mycorrhizal fungi. In general, we identify 72 genera differentially represented between soil and root microbiome. We also identified several fungal taxa which contain potential winter pea pathogens which are differentially represented across the locations. We found that both Didymella and Alternaria were highly abundant in one location in Garfield compared to other location, which also correlated with disease indices. We found significantly higher disease development in Garfield locations, which could be explained by the presence of Didymella taxa as well as Alternaria. Next, we identified fungal genera which were differentially represented between nodule and root microbiomes. We identify a number of these genera which were represented on a figure at the panel A. Most interesting aspect of this analysis was the identification of two yeast taxa, Cryptococcus and Salicocazyma, as well as Sclerotiniaceae family, which was specifically enriched in the nodules. This, you can see that these three taxa were highly abundant in the nodules. So in the panel B, shown the most abundant genera identified in the nodules. That was quite interesting. So next step was to apply PacBio long-read sequencing to narrow down the specific fungi which correspond to these ASVs. Our data showed that this yeast belonged to Cryptococcus flavus and Salicozyma area. Additionally, the Sclerotiniaceae was identified as Sclerotinia trifoliorum. Sclerotinia trifoliorum is a known P pathogen. However, very little is known of the presence of this pathogen in the Pacific Northwest soils. We didn't notice any significant disease development in the nodules while we were collecting samples. However, we didn't specifically focus on the diseases in the nodules. So next time in the future, it would be really important to assess the nodules for the diseases. Well, identification of Sclerotinia trifoliarum is quite interesting because it might be a pathogen we specifically target winter pea nodule tissue, and this infection might be very important in the Rhizobium legume association. For example, it might interfere with the nodule formation or interfere with the proper function of the nitrogen fixation inside the nodules. However, the actual role of this pathogen in the symbiosis needs to be investigated further, and right now we're in the process of isolation of this pathogen from the nodule tissue. Recent study identified some pectinolytic potential in several Cryptococcus flavius strains. It was shown that this yeast can exist inside the plant tissue and can have some plant growth promotion effect on the host plant. Interestingly, especially in respect with cold-tolerant winter pea, several Cryptococcus flavus strains exhibit strong cold tolerance. Similarly, several studies showed that Salicocazyma aurea exhibit a plant growth promotion function. It can secrete volatile organic compounds, synthesize indole acetic acid, and have ACC deaminase activity. Several Salicozyma strains also exhibit cold tolerance. Both of these yeast might play an important role in the adaptation of nodules to cold environments during winter and early spring, during formation of symbiosis between rhizobia and winter pea. In conclusion, multiple putative plant pathogenic taxa known to infect pea were identified in our study. The abundance of some of these taxa differ in the soils, root, and nodules across location. Regardless of relocation, the root microbiome was enriched with Rhizoctonia solani. The nodule microbiome was overpopulated with Sclerotinia trifoliarum, indicating potential susceptibility of nodule tissue to this pathogen. And finally, genera Cryptococcus and Salicocazyma were significantly enriched in the nodules compared to roots. These findings suggest the role of these psychotrophic fungi in symbiotic host fungal interaction. And finally, I would like to thank people who were involved in this research, Rebecca McGee and Lyndon Porter, my colleagues from USDA Agricultural Research Services, Alexis, Amber, and Jennifer, our lab technicians, and some collaborators from University of Idaho, Washington State University, and USDA ARS, Oregon. Thank you very much, and I'm ready to answer the questions.